Hi everyone, this is going to be our second lecture on the immune system. Previously we discussed the innate side of the immune system, and now we're going to start talking about the adaptive side of the immune system. Now this all covers the second portion of chapter 21. Remember, the first portion of chapter 21 was lymphatic, and there was not a lecture over that. We are only lecturing over the second portion of chapter 21. And before we start talking about adaptive immune, let's start with our not really attendance questions. Where does leukopoiesis occur? How does blood lost to the tissues during capillary fluid exchange make it back into the bloodstream? And are there more afferent vessels entering a lymph node or efferent vessels leaving a lymph node? So go ahead and try to answer those before watching to see what the answers are. And let's see, where does leukopoiesis occur? And before we can talk about that, let's talk about what leukopoiesis is. Remember, leukopoiesis is formation of leukocytes, white blood cells, and that is a part of hematopoiesis, formation of blood. Now, all parts of hematopoiesis, including leukopoiesis, occur in the red bone marrow. So that's the answer to this. How does blood lost to the tissues during capillary fluid exchange make it back into the bloodstream? Well, if you read chapter 21, or the first part of chapter 21, the lymphatic chapter, or if you remember earlier in the semester when we talked about capillary fluid exchange, you know that during that fluid exchange at the arteriolar end, fluid is leaving to supply the tissues. At the venular end, fluid is coming back into the bloodstream at the capillaries to bring waste in from the tissue so that it can be carried off. But also remember that all of the fluid that leaves does not get reabsorbed. Some of that fluid remains out in the tissues. And that fluid has to make it back into the bloodstream, otherwise our blood would dehydrate. Now, that fluid that leaves the blood capillaries actually makes it into what we call lymphatic capillaries. These are just very, very small lymph vessels. And that fluid is now called lymph. And the lymph moves through progressively larger and larger lymphatic vessels until eventually it's returned to the bloodstream up at the upper part of the chest, lower part of the neck, where the subclavian veins and the jugular veins meet. Now, are there more afferent vessels entering a lymph node or efferent vessels leaving a lymph node? So as that fluid, that lymph, is passing through all of those lymph vessels, it passes through at least one, typically multiple lymph nodes, little swellings. And you can see the diagram of a lymph node on page 810 during that self-study part of this chapter. Efferent vessels are the vessels entering the lymph node, bringing lymph from the tissues. And that was the afferent vessels. I don't know what I said. The afferent vessels are entering, and the efferent vessels are leaving the lymph node. Now, there are many afferent vessels entering a lymph node, but there are only one or maybe just a couple of efferent vessels leaving the lymph node. And what this does is it creates a backup. It makes that lymph remain in the lymph node for an extended period of time. What this does is it allows all of the white blood cells that are in that lymph node hanging out, it gives them a chance to inspect that lymph. Well, why would we want to inspect that lymph? Well, remember that fluid that left the bloodstream into the tissues? Well, when it's there in the tissues and it starts to move into those lymphatic capillaries, it's going to bring a sampling of what's going on in those tissues. Maybe you have an infection, maybe there are some bacteria, maybe there are some viruses, maybe there's something in that tissue, and that will be moved into those lymph vessels and eventually be carried into a lymph node. And then the white blood cells can say, hey, this just came from some of the tissues, there's probably more of them. Because if there is a bacterium in your body, there are many bacteria in your body. So this just kind of is a warning to get your immune system ready. And that's because there are multiple inputs, afferent vessels, and only one or a couple of outputs, efferent vessels, that cause that lymph to remain in the nodes for an extended period of time. Now we're going to start talking about adaptive defenses, the adaptive side of the immune system. And really, most of today's lecture is going to be very, very general information that just talks about how this part of the immune system works. We're going to start introducing a lot of terms, and then at the end of this lecture and all through the next lecture, we're going to come back and talk about those things that we're introducing now, but in more detail. So up until we start talking about how T cells work near the end of this lecture, if something seems a little bit confusing, don't worry, because we're only introducing it in a very general sense now, and we will come back and talk about it in more detail throughout the last part of this lecture and all of the next lecture. So the adaptive side of your immune system deals with very specific pathogens. In the previous lecture, when we talked about the innate immune system, we said that it is concerned with entry or spread of any pathogen. So it's not specific. It just knows this does not belong in the body. And something that results from that is while it's not specific, every single time that something shows up, whether it's the first time or the 12th time, your body treats it as though it is the first time. It has no memory of that pathogen. But the adaptive immune system, even though it's more specific, which may sound like a bad thing at first, it's not. Here, we're going to deal with, for example, flu type H1N1 of a certain strain, or a specific strain of streptococcus that causes strep throat. And while we're being very specific, we first have to actually be exposed to this pathogen before we can develop memory. So just like with a person, you can't remember something until it happens to you. Well, your adaptive immune system can't develop memory until it encounters the pathogen. And that will make more sense again once we start talking in more detail later. And the adaptive part of the immune system, just like the innate side had two branches, the first line and second line of defense, well, the adaptive, which is sometimes called the third line of defense, also has two parts to it. First, we have something called cell-mediated immunity, which deals with cells that are infected either by virus or certain bacteria, or by cancer, or we will see also transplanted cells. But in all of these cases, it's something within the cell is the problem. And the other half of the adaptive immune system is humoral immunity. And humoral immunity deals with things that are in your body's humors. Now, humors is an old word that we don't really use anymore, but it was a word that meant the liquids within the body. Now, even though we don't use the word humors anymore to mean that, we do still talk about humoral immunity because these are things that are within your body but have not yet entered cells. So if there's a bacteria in your interstitial fluid, the area in between your cells, or in your blood, which hopefully that's not the case because bacteria in your blood is its own really bad problem. But if it's something like a bacteria that is in your body but outside of your cells, then that's the area patrolled by humoral immunity. And we will talk in detail about both of these types. Now, in the PowerPoint that's available to you and in your outline, this part is next. I'm actually going to put this off, and in the next lecture, when we talk about B cell activity, we're going to come back and talk about this. So for right now, we're just going to skip over this part of the outline. Now, when we're talking about the adaptive immune system, we're really going to be talking about antigens a lot. So we mentioned antigens back when we were talking about blood types, but we're going to talk so much more about antigens now, and in much more detail. So first, let's just see what an antigen is. An antigen, by definition, is any substance that can provoke an immune response. And it's going to come down to shape. So antigens all have shape. And typically when we talk about antigens, we're usually talking about something on the surface of a cell or a virus. Now, they don't have to be, but typically we're talking about substances on the surface of a cell or a virus. And when they're something on the surface of a cell or a virus, they are either going to be proteins, carbohydrate chains, or a combination of the two. So a lot of times when we see the word antigen in a textbook, sometimes it's used to just mean something that's out of place in your body, but that's not really true. All of your cells have antigens on them, but so do all bacteria and all viruses and dog cells and tree cells, and everything has antigens on its surface. So let's be a little bit more specific. All of your cells have antigens, so we're going to call those self-antigens. Self-antigens. And then any antigen that is not your own is going to be called non-self antigen or foreign antigen. And again, this is going to be distinguished by shape. So your immune system is constantly checking every surface in your body and seeing what shape are these antigens. Hopefully, they're shaped like your self-antigens and everything is A-OK, and your immune system does not respond to them. It does not recognize them, which we'll talk about in just a moment. But if something has the wrong shape, that means it is a non-self or a foreign antigen. So it's all about shape of these antigens. Now, non-self antigens are recognized by your immune system, and it provokes an immune response. Now, something else that we're going to talk about are things called major histocompatibility complexes, MHCs. Major histocompatibility complexes. These are self-antigens, and they are a specific type of self-antigen. Now, we just said that self-antigens should not be recognized by your immune system. But MHCs, which we're going to talk about later on, MHCs actually are recognized by parts of your immune system. They do not look out of place, but they are recognized by your immune system. So we'll see what that means a little later on also. And all of this The shape of antigens and self versus non-self is the foundation for adaptive immunity. And over here, we can see kind of a cartoonized virus. And all of these little things sticking off the surface are proteins, and these proteins are the antigens of that virus. If there was a different virus there, it would have different antigens, and they would have slightly different shapes. Now, it doesn't have to be very different at all. The smallest difference means that it is a different antigen. We're also going to talk about antibodies. Antibodies are actually going to be a big topic of our next lecture, but we want to go ahead and introduce them here. We did briefly mention antibodies when we were talking about blood types. And we also saw when we were talking about plasma, another name for antibodies was gamma globulins. And antibodies are in your textbook on page 824 is where you can find this picture. And it goes into a lot of detail about the different types and the different regions of an antibody. You don't need to know all that. I do not expect you to know the different types. I do not expect you to know the different regions, but I do want to talk about how antibodies work very briefly here and in more detail in our next lecture. So antibodies are these branched structures. We saw before that usually they're shaped kind of like a Y. Sometimes they resemble more like a snowflake, or sometimes they resemble two Ys stuck together, but they're always branched. And at the ends of the branches, there's going to be certain shapes on this, it's called the binding site, or sometimes they're just called receptors. But they're going to have certain shapes here. And these shapes are typically shaped to fit against something that does not belong in your body. And when something that does not belong in your body that has a shape that fits this binding site, as we saw back in agglutination, they stick together. This binding site will stick like a puzzle piece to whatever antigen it is specific for. So that's how antibodies work. We'll see what results from that sticking later on in the next lecture. So let's talk about some of the cells that are involved in the adaptive immune response. We're going to talk a lot about lymphocytes. Remember lymphocytes from back when we were talking about the white blood cells in the first chapter? And we also talked about natural killer cells in the innate part of the immune system. Natural killer cells were a type of lymphocyte. Well, now we're going to look at some other types of lymphocytes. First, we're going to talk about T lymphocytes, which are sometimes just called T cells. Now, you may have heard of T cells and B cells before, but let's see a little bit more about T cells. So T lymphocytes, like every blood cell, originates in the red bone marrow. But now when we talk about T lymphocytes and B lymphocytes, even though they're made in the red bone marrow, once they're made, they're actually not ready to do their job yet. We say that they are immature as soon as they are finished being made. So they have to go through a process called maturation. And maturation occurs in different places around the body. T lymphocytes, for example, they leave the red bone marrow after they're made when they're still immature, and they travel to an organ called the thymus. The thymus. Now, the thymus is a gland in the lower part of the lower part of the neck, the upper part of the chest. And it's weird in that it's the only organ that's bigger when you're born than when you're an adult. It shrinks throughout your life. But the thymus is where T cells go to mature. So what does mature mean when we're talking about cells? Well, B lymphocytes and T lymphocytes, we just said when they're first made, they're not ready to do their job. So how do we know if they're ready to do their job? They have to pass some tests. Now, T lymphocytes, when they get to the thymus, they are put to a test for immunocompetence. Immunocompetence. And what that means is every single T cell has receptors on its surface. And those receptors are shaped to fit a very specific foreign antigen. Every T cell will have different receptors for different foreign antigens. So every T cell has a specific foreign antigen that it's looking for. Immunocompetence tests to see, does this cell respond to its specific foreign antigen? We want it to recognize and have a reaction when it's going to meet its specific foreign antigen. If it does recognize its specific foreign antigen, and if it responds to it, then it passes the test and it moves to the next step. But if it does not recognize and does not respond to its specific foreign antigen, it does not have immunocompetence. So it's going to be eliminated through something called positive selection. So that cell that did not pass immunocompetence testing is going to be destroyed because it's useless. It can't do its job. There's no reason for it to continue. Now the cells that passed through the positive selection part, that are immunocompetent, move on to the next step. They're going to be tested for self-tolerance. Self-tolerance is going to test to see, does this cell not recognize, does it not respond to self-antigens? We do not want it to respond to self-antigens. It should not recognize them. If it does recognize them, it's going to be destroyed through a process called negative selection. The cells that do not recognize self-antigens, that do not respond to self-antigens, pass, and we say they are self-tolerant. Once a cell passes immunocompetence testing and self-tolerance testing, it's ready to go. It is fully mature. So what does it do when it's fully mature? It leaves the thymus and it goes out into circulation. It's going to circulate in the blood. It's going to circulate through the lymph, but mostly it's going to hang out in the lymph nodes. It's just going to sit there in the lymph nodes most of the time. Sometimes it's going to leave and go into circulation. Sometimes it's going to leave and wander through the lymph system or the cardiovascular system. It's going to circulate, but eventually it's going to come back and just kind of hang out in the lymph nodes again. So at this point, we say that this T cell is naive. We'll come back to it in just a little bit. But remember, when it's first made, it's immature. It leaves the red bone marrow to go to the thymus. Now it's going to be put through those tests, and it becomes mature, but it is naive. And we'll see what that means here in just a moment. But let's look at B cells. B lymphocytes, just like T lymphocytes, just like every other blood cell, originates in the red bone marrow. But it remains in the red bone marrow where it's going to go through maturation. Now, B cell maturation, we don't know too much about. We believe it's pretty much the same as T cell, where it's going to pass through the positive and negative selection, the immunocompetence and self-tolerance testing. But we're not completely sure. But again, once the B cells have matured, they are naive, they go out into circulation, but most of the time, they're hanging out in the lymph nodes. So the B cells and the T cells, they've matured, they are naive, they're probably hanging out in a lymph node. They may be circulating for a little bit, but eventually they'll come back and hang out in lymph nodes again. So what does naive mean? Naive means it's been made, it has achieved maturity, and it has left to go into circulation, but it has not yet encountered its specific foreign antigen. Naive cells are fully mature, but they have not yet encountered its specific foreign antigen. So, where does that antigen encounter occur, if it occurs? It could encounter it anywhere, but chances are it's going to happen in a lymph node because that's where the cells are most of the time. But it could happen anywhere in the body. And what happens when it encounters its specific foreign antigen? Well, that is when we start to have a fight. So, let's see B cells and T cells. Now, they're going to follow a common theme. Each B cell is going to follow a common theme. Each T cell is going to follow a common theme. B cells and T cells together are going to follow a common theme, but they're going to do it a little bit different. So let's just see what that common theme is. Well, here we have a B cell or a T cell that is naive. Could be either a B cell or a T cell. And remember when we were talking about formed elements way back when? Something that they all had in common was they were mostly amitotic. They did not go through mitosis. Well, these cells do go through mitosis, but they only do it if they have been activated by encountering their specific foreign antigen. But even though they are going to go through mitosis, it's a very specialized mitosis. Remember cell division, mitosis? When a cell divides, it produces two cells that are identical to each other and to the cell that we started with. When we're talking B cells and T cells, there's a little bit of a twist on it. They're going to produce something called clones, but they're not all identical. They're going to go through a process called differentiation. So, let's see what that means. Here is our naive cell, and our naive cell is going to encounter its specific foreign antigen. It becomes activated, and now it's going to start to divide. It's going to differentiate. And it's going to differentiate into two cells that are similar to the cell that we started with, but not identical to the cell that we started with. So here's the cell that we started with. It divides into two similar cells, but they're not identical. Over here on the left, this is going to be something called a memory cell. And over here on the right is called an effector cell. And now the memory cells are going to start to go through proliferation. And this is pretty much the same as mitosis. We're going to make lots of memory cells. The effector cells are going to do the same thing. They're going to go through proliferation and make lots of effector cells. So what's the difference between memory cells and effector cells? Well, memory cells, they don't do anything right now. They actually go and hide in your lymph system. The effector cells are the ones that are going to fight this current fight. Now, B cells and T cells will fight differently. So we can't really say how the fight happens, but the effector cells are going to fight the current infection. Now, there's a little bit of something we want to go back and talk about with the memory cells. Even though they do not fight this current fight, they are ready to fight in the future. What does that mean? Well, this process from naive all the way down to many effector cells takes almost 2 weeks. It takes, between a week and 2 weeks. And during that time, typically you feel pretty miserable. And that's before your effector cells are actually ready to fight. Now, memory cells, they go hide. They don't do anything at all during this fight. But let's say next year, that same pathogen shows up again. Well, these memory cells are going to recognize it because now we have memory. And these cells immediately convert to effector cells. And they start fighting right away in a matter of a few hours to a couple of days at most. And you do not get sick that second time, or third time, or fourth time. So memory cells don't do anything now, but they're ready to swing into action if something ever comes back. So again, right now, all we're doing is introducing just the groundwork. We will come back to all of this and look at it in much more detail later. So some other cells of the adaptive immune response are antigen-presenting cells, or APCs. Antigen-presenting cells are not a part of the adaptive immune system. They're not specific. These are things like macrophages, neutrophils, things like that. Things that are non-specific. They use things like those pattern recognition receptors that we talked about last time. But they play an auxiliary role in cell-mediated immunity. What does that mean? Well, when one of these phagocytes comes along and eats a cell, it's actually going to go and tell your T cells that something is here that shouldn't be. So it helps your adaptive immune response. It is not a part of your adaptive immune response, but it helps your adaptive immune response. And it helps using those major histocompatibility complexes that we talked about a moment ago. So these cells, they're phagocytes. Now, remember back in Bio 137 when we talked about phagocytosis? We're going to talk about it again now, but we're going to introduce some more detail with how phagocytosis actually works. So over here on the left and here in the middle, we see phagocytosis in action. And over here on the right, we see an image of phagocytosis in action. Or actually, this isn't really phagocytosis. This is a cell that has phagocytized something, but is talking to a helper T cell like we just saw a moment ago. But up here, these are the steps of phagocytosis. Now, this green thing is some sort of pathogen. I'm going to call it a bacteria just for purposes of this example. This big purple thing is some sort of phagocytic cell. I'm going to call it a macrophage for this example. Now, first, this macrophage is going to use pattern recognition receptors to identify this bacteria as foreign. Now, remember opsonization? Maybe opsonization helped this process. Maybe not. This will still occur whether or not opsonization occurred. But once this bacteria is recognized as foreign, this macrophage will start to extend pieces of its cytoplasm outward. These are called pseudopods. It's going to start to extend pieces of its cytoplasm outward. It's like it's giving a hug to the bacteria. But eventually, these pieces of cytoplasm are going to meet. And it's going to completely envelop this bacteria and enclose it in a vesicle. So this bacteria is now trapped inside of a vesicle. Remember, vesicles are just those sacs that contain something inside of a cell. Well, this vesicle contains the bacteria. But let's be more specific. Since this vesicle was formed by phagocytosis, we're going to give it a new name. We're going to call it a phagosome. The phagosome is just the vesicle that was formed by phagocytosis, and now it's containing that bacteria that we just grabbed. Well, now we have a bacteria inside the cell, we need to get rid of it. So how do we get rid of it? Well, there's another specialized vesicle we learned about in Bio 137 called a lysosome. A lysosome is kind of like the stomach of the cell. It's a vesicle that contains digestive enzymes. Now, the lysosome is going to come along and merge with the phagosome. It's actually going to become one really big vesicle that contains the bacteria that we just ate, all of those digestive enzymes, but all in one specialized vesicle. So let's give it a name. Phagosome plus lysosome equals phagolysosome. A phagolysosome is a vesicle that contains whatever we just phagocytized, in this case the bacteria, plus all of those digestive enzymes. So it's going to start to digest that bacteria. And it's going to break that bacteria down into really small pieces. Some of those pieces can actually be reused by the cell. Things like amino acids, things like lipids, the cell can use those. But the things that we can't use are going to be called the residual body. And the residual body is waste. It's kind of like poop for the cell. And it's going to be exocytosed. And that residual body is removed from the cell. Eventually it makes its way to the bloodstream. It circulates to the kidneys. The kidneys will remove it from the blood, and you will urinate it out. But we have to show that we had this bacteria show up in the body, because if there is one bacterium in the body, there are many of that bacteria in the body. So this guy just ate the evidence. How do we inform the T cells of what we just ate? Well, down here, when this bacteria is being broken down, what actually happens is we take some of its antigens, some of those pieces that show what it looks like, and we move it out onto the cell surface on one of those MHCs that we talked about, the major histocompatibility complex. So now this macrophage is wearing a piece of its dinner, kind of like a hat. It's saying, hey, look what I ate. And is going to go and show your adaptive immune system, hey, I just ate something. This is what part of it looked like. Now go look for it. So that's how phagocytosis and MHCs are going to work together. So the rest of this lecture is going to be focused on part of cellular immunity, the T cell side of things. And like we said earlier, T cells, cellular immunity, are going to focus on cells that have something wrong with them. So this could be infected cells, cells that either have a virus or certain types of bacteria, cancerous cells. Now, virus-infected cells and cancerous cells, what they have in common is that once they are virus-infected, they are forever virus-infected. Remember when we were talking about interferons? Also, cancerous cells, all a cancerous cell is a cell that's going through mitosis when it shouldn't be. But the thing about cancerous cells is when they divide, they make 2 cancerous cells, and then those 2 make 4 cancerous cells, and they keep dividing. And once a cell becomes cancerous, it is forever cancerous. So infected cells and cancerous cells, there is no fixing them. And transplant cells, for purposes of our body and our immune system, they kind of fit that same category. Now we know that we get transplants to help us. They're for a good cause, but our immune system doesn't know that. To our immune system, a transplanted cell just looks like a cell that is out of place or has something wrong with it. So it falls into the same category. These are the cell types that will be dealt with by cellular immunity and T cells. But when we're talking about T cells, there's actually many different types of T cells. We're going to talk about the big 3. And unfortunately with T cells, the naming can get a little bit confusing. So let's look at the different names that we will be using. The first type is going to be, when it is naive, it's going to be called a CD4 cell. A CD4 cell is a naive version, but when this version encounters its specific foreign antigen and it is activated, it goes through a little bit of a change, and we call it a helper T cell. Sometimes it's written T with a little H next to it. And helper T cells have a very specific reaction when they're activated. We'll see what in just a little bit. So how does this name come about? Well, CD4 is because the receptor on the surface of the cell is called a CD4 receptor. The next type is a CD8 cell, named because of its receptor. Now, CD8 cells are naive. When they become active, because they see their specific foreign antigen, they go through a change and they start to be called cytotoxic T cells, or T with a little bitty C next to it. So CD8 is the naive version. Cytotoxic T, or T sub C, is the active version. Now, down here, we've got 4 different things written, and they all 4 mean the exact same thing. Regulatory T cell, T sub reg, suppressor T cell, or T sup, those are all for the same thing. Different books call them different things, but they're all the same thing. So helper T cells do a specific job. Cytotoxic T cells do a specific job, and regulatory T cells do a specific job. And we're going to look at all 3 of them. Now, something about the cellular immunity, these T cells, that seems a little bizarre is that they must have the antigen presented to them. They don't do anything with free antigens. If they were to circulate through and come up on a free antigen that's just floating there, it does absolutely nothing. They actually have to have the antigen shown to them or presented to them before they can recognize it. Now, those MHCs that we were talking about are going to play a role in cellular immunity. Remember, MHCs, they are self-antigens. They are just a specific type of self-antigen. And there are different types of MHCs. We're going to talk about MHC class 1 and MHC class 2. They have slightly different functions. They're similar, but slightly different. MHC class 1 is found on every nucleated cell in your body. So all of the real cells in your body that are not, like red blood cells, remember they don't have a nucleus. Thrombocytes, they don't have a nucleus. So all body cells that are true body cells have MHC class 1. MHC class 2 is found on antigen-presenting cells, and that's all. So class 1, class 2, they're going to have similar function, but there's a little bit of difference, and they are found on different types of body cells. So next what we're going to do is draw the activities of T cells. We're going to draw helper T cell first, and then that's going to be the end of this lecture. Cytotoxic T, regulatory T, those are going to be in the next lecture. So let's go ahead and look at how helper T cells work. Okay, so let's look to see how helper T cell activity works. Now what we have up here at the top are various CD4 cells. Remember, CD4 are the naive version of helper T cells. And so we have the cell, and then this is the CD4 receptor sticking off of it. And at the end of each receptor, here is its specific shaped region so that it can recognize a very specific foreign antigen. And it's going to do that by fitting this end of the receptor against its antigen. If it fits, that shows, hey, this doesn't belong here. If it doesn't fit, then nothing happens. It doesn't recognize it. So what I've got drawn here is a very exaggeratedly large bacteria. Really, bacteria are much smaller than body cells. But here is a bacteria, and then these little things sticking off of it are its antigens. So these are foreign antigens. And what we're drawing here could happen anywhere in the body, but chances are what we're drawing is happening in a lymph node. So first let's start with this guy with the squiggly shape receptor. He comes along and they are touching their receptor against every surface in your body. And he touches his squiggly shape receptor up against that bacterial antigen and it doesn't fit, so he doesn't recognize it. And he moves on, still naive. And then square shape receptor. Same thing. He bumps his CD4 receptor up against that bacterial antigen. It doesn't fit, so nothing happens, and he leaves, still naive. And that happens, countless times. I've only drawn 4, but there are an endless number of shapes. Eventually, round shape receptor is going to come along and bump his receptor up against the antigen, and it is a perfect fit. But nothing happens. He is not activated. He leaves, still naive. So why did nothing happen? It fit. Well, remember, T cells have to have the antigen presented to them. They don't recognize free antigen, which is what this is. So eventually along comes macrophage. Phagocytosis occurs because this macrophage uses his pattern recognition receptors and says, hey, this doesn't belong here. It looks like it's probably some sort of bacteria, and he consumes this bacteria. Phagocytosis occurs. And he gets the goods that he wants. The macrophage does. He uses some of those amino acids for himself. The residual body is kind of popped out through exocytosis. But on the surface is an MHC class 2. Remember, the MHC class 2 is on the surface of antigen-presenting cells, which macrophages are. And after this macrophage consumes that bacteria, he moves a piece of the bacterial foreign antigen out onto the end of that MHC class 2. And now, eventually, square shape receptor is going to come along and touch and not recognize it and leave. And again, triangle shape receptor, same thing, comes along and he leaves. Eventually, let me draw that a different color. Eventually, round shape receptor is going to come along. And some very specific things have to happen now. So this receptor, it has this other arm. And this other arm is going to be very necessary for the next few things to happen. First, this arm is what reaches around and feels MHC class 2. And that has to happen first. This arm kind of gives this little reach around here and says, hey, you're an MHC class 2, you're probably going to show me something, so let's see what it is. And that starts the activation process. But there's still a lot to go. Once the arm feels MHC class 2, this other piece of the receptor reaches in and fits, and these two pieces come together and it is a match. So step 2, now this T cell starts to become activated. He's not fully activated yet, but these two steps started the activation process. The reach around arm says, hey, you're going to show me something, aren't you? The end of the receptor says, oh, that's what you've seen. I should probably do something about that. So this cell is getting ready to become activated. Now, meanwhile, the macrophage feels himself getting kind of, examined, kind of felt up there a little bit. And he's, oh, you're checking me out, huh? And he sprays a chemical called interleukin-1. Interleukin-1 is released by the macrophage when a helper T cell that is inactive is docking for inspection. When a naive helper T cell docks for inspection, the macrophage, or the antigen presenting cell, releases interleukin-1 onto the helper T cell. And that is the chemical that finalizes the activation process. So once all three of those things have happened, the reach around arm feels MHC class 2, the receptor end recognizes the specific foreign antigen, and macrophage, or antigen presenting cell, releases interleukin-1 onto him. Those three things result in activation of this CD4 cell. And what happens next is we're going to go through clonal selection. We're going to go through that proliferation and differentiation stuff. We're going to first produce memory cells. Those cells go hide. So these are memory helper T cells. Your book kind of is misleading. It only says that cytotoxic T cells produce memory cells. That's not true. Helper T cells also produce memory cells. So we have memory helper T cells. They go hide. But remember, they're ready to fight in the future. They're just not going to do anything right now. And we make active helper T cells. So we're just going to go ahead and call these T helper cells, or helper T cells. These are the effector cells that are produced from CD4 cells. Remember, memory cells don't do anything now. Effector cells fight the fight. Well, when we're talking about CD4 and helper T cells are the effector cells. That's their name. So what do active helper T cells do? They release a chemical called interleukin 2. And interleukin 2 is kind of like an alarm call. Interleukin 2 tells all of the other cells in your immune system, hey, there is something here that we need to fight, so get into fight mode. The helper T cell is kind of like the tattletale. He's running along and telling everyone get ready to fight. Now, he doesn't actually fight himself. He's active, but he's not doing any fighting. He's just telling everyone else to get ready to fight. And he does that with interleukin 2. Interleukin 2 is a chemical that sounds the alarm that the body needs to prepare for a fight. And without interleukin 2, the rest of your immune system doesn't really do anything. So helper T cells, it may seem like they're not really doing much because they're not fighting. But we're going to come back to that. That phrase that I just used is going to be really important. Interleukin 2 is what tells the rest of your immune system that a fight is here. And while helper T cells don't fight, they actually are responsible for the rest of your immune system going into fight mode. All right, so that's where we will stop with this lecture. The next lecture, we're going to talk about the other two types of T cells. We're going to talk about B cells, and we're going to kind of wrap everything up with the immune system. All right, I will talk to you next time, but let me know if you have any questions.